genetic condition has liver component, and the Gaucher is definitely a fascinating disease for research understanding pathophysiology wise. And so he's, as I mentioned, he's active for both, and he also was the former chief of the section of the hepatology and the pediatric hepatology before. So that's a good connection for the pediatric population too. So with that kind of short introduction, I'm letting him to take the floor. Thank you again, Dr. Mistry, for presenting to this our groundwork. So.
Thank you, Dr. Jiang. It really is a great honor to be invited to give this grand round. And you have kindly allowed me to talk about on this particular topic, which is neuronopathic Gaucher disease. This is a huge unmet need. And in fact, if you look at the world population, there are many times more neuronopathic Gaucher disease cases than type 1. But the world literature is entirely focused on very European-centric, N370S-centric Gaucher disease, which really arose in the Eastern Europe through a founder mutation. So these are my disclosures. I have research grant from Sanofi, and I've received travel support from them. So members of Department of Genetics really don't need much introduction to Gaucher disease. It really is in so many ways a prototype inborn error of metabolism and a prototype lysosomal disease due to deficiency of this enzyme, acid beta-glucosidase, due to biallelic mutations in GBA. And there are three broad categories of Gaucher disease. One is type 1, and for us in the United States and the Northeast particularly, this is the most important one where there is no early onset of neurodegeneration. And it is pan-ethnic, but it is very prevalent among people of Ashkenazi Jewish ancestry, affecting about 1 in 850 individuals. And the big clinical challenge is that it can have an onset in childhood or even into late adulthood, first-time patient presenting with AVN in their 70s. And this type also has 26-fold increased risk of Parkinson's disease, Lewy body dementia, compared to the normal population. And then there is the fulminant type 2 disease. This is what in genetics we refer to as collodion babies, which is the most dramatic form of type 2. And then often we have fetal death at birth or intrauterine and hydrops fetalis. But some of these babies are being born, and you see neuronopathic symptoms very early within the first few months of life. And then there is a type 3 where there is a huge spectrum. I have type 3 patients who have IQ of 140 and have autism type disorder. That is the only neuronopathic manifestations in such individuals. So work that we have done in our lab, led by Shiny Nair, research scientists and others, have shown that glucosylceramide metabolism spans multiple cellular compartments involving the Golgi, lysosome, and cytoplasm. And our recent work is also starting to implicate mitochondria. So glucosylceramide is a very important lipid for cells, and it is synthesized via glucosylceramide synthase enzyme in the Golgi. And as membranes turn over, particularly in the macrophage system, the lipid is regenerated from breakdown, stepwise degradation of complex glycosphingolipids. And so when glucosylceramide enzyme is deficient due to biallelic mutation, this lipid accumulates. And as it accumulates, an alternative metabolic pathway kicks in via acid ceramidase enzyme which deacylates this lipid to glucosyl sphingosine, removes an acyl group, and that allows this lipid to be exported out to the lysosome, to the cytosol, and into the plasma. And as I'll show you, in fact, this is now an exquisite biomarker of Gaucher disease. These two lipids are building up in circulation and in cells, glucosylceramide and glucosyl sphingosine, are highly immunoactive and inflammatory, and they trigger B-cell lymphoproliferation and metabolic inflammation, wide spectrum of bone disease, as well as neurodegeneration. And what I'm going to share with you today is neuroinflammation, how important that is in neurodegeneration. And so this is the pathway that has been delineated by really pioneering work of Shaini Nair in my lab. And what she found in a volley of papers is that glucosylceramide accumulates in the macrophages and antigen-presenting cells, and glucosylceramide and its downstream product, glucosyl sphingosine, are presented in the context of CD1 molecules. And this complex is recognized by T cell receptors on type 2 NKT cells, which have follicular helper phenotype, and that allows this cell type to then lead to germinal cell B-cell proliferation and plasma cells which are producing anti-lipid antibodies. And these anti-lipid antibodies then activate the complement, and the complement activation product C5a then interacts back in the tissues to the C5a receptor. And any sterile inflammation, metabolic inflammation, one of its downstream effects is induction of glucosylceramide synthase, making more of the lipid that is already building up in Gaucher disease. And so there is a positive feedback loop which amplifies the underlying enzymatic defect due to genetic defects in GBA enzyme. And so that tells us that therapeutic targets are to try and give the enzyme back either via recombinant enzyme or via gene therapy, that is a focus of a lot of attention. But the other is that GCS appears to be an exquisite therapeutic target. So for a long time, the focus of Gaucher disease investigations and therapeutic endeavors have been on the macrophage. And that has been very fruitful because the macrophages that are lipid-laden produce a variety of molecules listed here, GPNMB, chitotriosidase, CCL18. And these are now biomarkers of Gaucher disease, and we use it routinely in monitoring patients with Gaucher disease. But the other thing is that these macrophages exhibit a macrophage Menos receptor, and that is how recombinant enzyme is targeted to the lipid-laden macrophages in the body. And this gamma CT scan imaging of a tracer dose of I123 label Menos-terminated enzyme, only 150 nanograms, and the patient is on a gamma CT scanning camera in nuclear medicine, and you can see enzyme molecules going in, and within the 20 seconds, it's already in the spleen, and you can start to see it appear in the liver, and this is exquisite targeting to the sites of disease activity, and also to some extent in the bone marrow. So this is work that really was the outcome of nearly a quarter of a century of sustained efforts by a Lasker Award awardee, Dr. Roscoe Brady and his team, that really led to a transformation in how we care for patients with Gaucher disease. So as I mentioned, glucosylceramide metabolism is compartmentalized. There is the synthetic pathway, and there is the catabolic pathway. And the therapeutic targets are directed either to replacing the deficient enzyme in the lysosome or reducing the synthesis via inhibitors of glucosylceramide synthase. So these inhibitors, you might argue, are like statins of glycosylphosphate world. So the classical phenotype descriptions of Gaucher disease that I alluded to are type 1, type 2, and type 3. And the distinctive feature of type 1 is that there is no childhood onset neurodegeneration. And the signature hallmark of type 1 is that patients have at least one N370S allele, a founder mutation from Eastern Europe. And this is one place where genotype-phenotype correlation can be very helpful in genetic counseling, because if a baby has at least one N370S allele, you can absolutely guarantee to the patient, family, parents, that there will be no neuronopathic Gaucher disease. And as I mentioned, type 2 and type 3 are the very severe forms of Gaucher disease that have all the visceral hematological manifestations. But even among type 2, for example, there is a huge phenotype spectrum, from perinatal hydroxyphthalate and collodion skin to at newborn and to infancy. And type 3, again, there is a huge phenotype divergence. And so at one end, we can have a child age 1 or 2 years with severe saccades and failure to thrive and neurological disability. At the other extreme is like the patient that I take care of, who has an IQ of 140 with autism spectrum disorder. But type 3C is a particularly interesting one. This is always due to N370S D409H mutation, homozygosity. And it is like other forms of Gaucher disease, but it is uniquely associated with cardiac valvular, aortic valve calcification, and coronary artery disease. So these children die in their second decade of life, usually due to myocardial infarction and or aortic insufficiency. And this phenotype is consistent, whether you see a patient in Japan or in the United States or in the Middle East. And so this is probably the most consistent genotype-phenotype correlation, and it raises intriguing questions for translational research. So the GBA gene locus that codes for the defective enzyme in Gaucher disease is shown here, GBA1, with the two common mutations, N370 as the founder mutation, and L44P, that homozygosity of which is associated with neuronopathic Gaucher disease. This is on chromosome 1q22. And this figure was kindly prepared for me by Jeff Luan in my lab. And you can see that in literally 150 kilobases of DNA, there is a very gene-dense region, including nearly a dozen genes and 2 pseudogenes. And what makes this locus so complicated is that the active gene is closely linked to a highly homologous pseudogenes, and the pseudogene harbor mutations, which if transferred to the active gene by gene conversion events, leads to Gaucher disease. And one example of that is the L44P, which normally occurs in the pseudogene, and worldwide, this is the most common mutation underlying Gaucher disease. And so there are two views of Gaucher disease, and sadly, the literature is 99% almost European-centric, related to N370S Gaucher disease, namely type 1, and this arose in the 11th century in Eastern Europe, and it has been widely disseminated. But as I mentioned, L44P is actually quantitatively many times more common worldwide, and it is the cause of the devastating types of neuronopathic Gaucher disease and a huge unmet need. And thankfully, we are now starting to focus. And that's what I want to talk about today in my presentation, neuronopathic forms of Gaucher disease, because it is always so heartbreaking when we see parents with their babies who have neuronopathic Gaucher disease, and there is very limited therapeutic options that we can offer these individuals. I mentioned D409H homozygous genotype, and this was first described in Japan and subsequently in Spain, as well as in Jerusalem, Israel. And this unique features which are not found in any other types of Gaucher disease. This is an area that we are very interested in. Unfortunately, the alpha-4p homozygous knock-in mice do not have this phenotype. So the reason it is so heartbreaking to see a baby with neuronopathic Gaucher disease is because they develop this kind of severe disability that can start just with failure to be able to sit up, and then saccades which lead to difficulty in tracking objects, and then it evolves over time into seizures, intellectual disability, and complete neurological disability. I think that we really must commit to understanding this type of Gaucher disease, and through that, to develop therapeutic approaches. So the major questions that relate to this important form of Gaucher disease is what is the functional significance of microglia GBA in neuronopathic Gaucher disease pathophysiology. As I mentioned to you, Dr. Nayer's work have shown that the lipids that accumulate in Gaucher disease in the systemic organs are highly inflammatory. What happens in the brain? There's a paucity of data on this. Most of the work have focused primarily on neuronal toxicity, but not on neuroinflammation. And the view has been that if there is neuroinflammation, then that is largely a response to injured neurons. We wanted to define the immune landscape in neuronopathic Gaucher disease and understand the role of GBA in non-neuronal cell types, and more importantly, discover biomarkers. And this is very important because when we think about clinical trials, we are recruiting people who have already manifesting neuronopathic disease. So these individuals already have significant brain injury and have saccades and so on. And I think that there is an urgent need to identify biomarkers of neurodegeneration that will allow us to harness the greatest potential of therapies. And then, of course, whether brain penetrant therapies such as GCN inhibitors, can they abrogate dyshomeostasis of glucosylceramide metabolism and immune dysregulation. And just a few weeks ago, we published this paper, and it was a gigantic piece of work. And I want to credit our colleagues here at Yale and members of my lab, and they worked incredibly hard through the pandemic to bring this study together. So what we have done is we've taken neuronopathic Gaucher disease mice. So this enzyme is very important in the skin, and its loss leads to collodion baby type syndrome, and the mice die immediately at birth and in utero. And so these mice are flocks, and they are crossed with a K14 Cree. And so the intact GBA is present in the skin, but all the cell types in the body are deficient in GBA, including astrocytes, neuron, and microglia. And we crossed these mice with CX3CR1 in order to rescue GBA in the microglia, to understand what its role is in the microglia. And we crossed these mice to nestin Cree in order to understand its role in the neurons. Then we had developed these mice, Flox GBA mice, and we crossed them with CX3CR1. And so in these mice, the only cell type really practically that's deficient in GBA is the microglia. And our hope was that studying these mice will allow us to really focus on the role of microglia in neuronopathic Gaucher disease, and perhaps it may even lead to bolstering data for putative biomarkers. The brain is, of course, very complex, and I think that we need these kind of strategies to deconstruct neuronopathic Gaucher disease. And then we did the standard process of immunophenotyping, lipidomics, single cell analysis, impact of GCS inhibitor, and then try to leverage our findings to discover biomarkers. And so in neuronopathic Gaucher disease, we found that there is a remarkable attrition of microglia. In the wild type mice, the main immune cells are the microglia, and then there are a variety of other cell types. But we find that in neuronopathic Gaucher disease, there is an attrition of microglia and a variety of other immune cells, and in particularly NK cells here are not present in wild type mice, but they are present in surprising numbers in neuronopathic Gaucher disease. To try and do lipidomics and immune on microglia and immune filtrates in these mice is difficult. You have to pool many mice to get enough cells for lipidomics, and my lab really did a fantastic job to accomplish that. And what they found was that neuronopathic Gaucher disease microglia accumulate glucosylceramide as well as glucosylsphingosine. So these microglia are lipid engorged. And moreover, the infiltrating immune cells shown here were also laden, particularly with the glucosylceramides, but not so much with glucosylsphingosine. This is a principal component analysis. And what we found is that microglia underwent an attrition. The microglia changed from homeostatic phenotype, which is the wild shown here in the wild type mice. And the wild type mice do not express this damage associated microglia genes. And these are the damage associated microglia that have been described in a variety of neurodegenerative diseases. And that pattern reverses in neuronopathic microglia, so that there is a downregulation of the homeostatic genes and an upregulation of the damage associated microglia genes. So not only is there an attrition of homeostatic microglia, there is a dramatic transformation in the gene transcription profile to damage associated microglia. And so these are the Tisney plots depicting different microglial and non-microglial subsets. And the clusters are coded based on mice affiliation on the left, as you can see here. And in total, there were some 14 clusters containing 6 microglia clusters and 9 clusters of immune cells that are shown on the right. And again, the transcriptome profiling showed that the homeostatic microglia undergo attrition, and there is a widespread dysregulation of immune cells in the setting of neuronopathic Gaucher disease. And so this is the hierarchical clustering of differentially expressed genes. And it shows that type 1 interferon genes from neuronopathic Gaucher disease are markedly increased compared to control mice. And all individual type 1 interferon genes with significant differential expression are shown here on the right, And it is a remarkable upregulation of so many of, in fact, the interferonopathy genes. So we were very interested in pursuing the NK cells. And indeed, when we did flow cytometry in NGD mice looking for NK cells, there was a marked increase in the neuronopathic Gaucher disease. And that was also the case in NGD mice in whom we had restored GBA in the microglia. And these NK cells were highly activated, indicated by this granzyme A positive cells. And so this may be one of the first kind of indication where there is potentially a role of NK cells in driving neurodegeneration in the setting of inborn error of metabolism. So when we rescue GBA in the microglia and the neurons, this is what we see on these survival plots. And this is a survival of the neuronopathic Gaucher disease mice. It's around 18 days. And after rescue in the microglia, we can significantly improve the survival. And when these mice have rescue in the neurons, they survive much longer, but at 200 days develop quite severe neurodegenerative disease and succumb to that. And in these different mice, we performed lipidomics, and these are the data on different species of glucosylceramide lipids. And you can see that there is a dramatic increase in glucosylceramide lipids in NGD mice, shown in green. And when we rescue GBA in the lipids, and there is a further reduction in the lipids when in the NGD mice we rescue GBA in the neurons. But the most important learning point of this capacity to metabolize this toxic lipids. So the microglia are based on this data. You can see that it can account for nearly 89% of the processing of these lipids, which has certainly been a huge surprise to me and my group. And when there is neuronal rescue, we can decrease the lipids by nearly 98%. So I mentioned why biomarkers are very important, because really to do the best we can for the patients and to really design powerful clinical trials, we should probably have biomarker-based inclusion criteria in endpoints. And right now we don't have that. The only biomarkers that are present are neurological symptoms such as seizures and saccades and so on. And so these are the mice that we looked at to try and start looking at what the biomarkers may be. And so these are the floxed mice in whom the only deficiency is in the microglia. And we looked at young and aged mice, and we find that in fact this neurofilament light chain is increasing, and it increases with age compared to wild type mice. And this remarkably is correlated with increase of glucosylsphingosine. So there are molecules in the brain that are clearly appearing in the circulation that is reflecting neurodegeneration. And so we were very, very excited. And again, in addition to this biochemical measurements, we found that GBA deficiency in the microglia did lead to age-related microglia activation, as well as increase in immune infiltrates. These data are quite important for the late onset neurodegeneration of Parkinson's disease, Lewy body dementia in type 1 Gaucher disease. And so what we did was to did single nuclear RNA sequencing on the brains and resolved the different cell types into 61 different gene clusters, as shown here, and the different major groupings in these mice. So these were the NGD mice, which were at 2 weeks, and these were the NGD mice in whom the microglia had been rescued for GBA at 2 weeks when they were healthy, and at 6 weeks when they were starting to exhibit neuronopathic disease. And so we wanted to look at the effect of aging in these mice. And another aspect of looking at aging was to examine these NGD nesting-free mice at 7 months. And these were the number of cells that were studied. And so this is the area AUC analysis for select lysosome interferon signature genes, chemokine, and ApoE gene sets that were significantly enriched in the microglia, Purkinje cells, oligodendrocytes, astrocytes, and neuronal clusters of NGD mice at 2 weeks versus control mice, and NGD CX3CR1-CRE at 2 and 6 weeks, respectively, versus the control mice, and the NGD nesting-free mice versus the control mice. And we were particularly interested in apolipoprotein E because of its role in lipid metabolism as well as in neurodegeneration. And normally the microglia exist in the astrocyte, and you can see a marked upregulation of microglia beyond the astrocytes, upregulation of ApoE beyond the astrocytes to microglia, Purkinje cells, oligodendrocyte, as well as the different neuronal population. So we wanted to pursue this vigorously, and because of the link of apolipoprotein E genotypes and neurodegeneration to Gaucher disease. And what we found on this single nuclear RNA sequencing, that all of these sets of mice have a marked upregulation of apolipoprotein E expression. And concomitantly, ABC1, which works in concert with apolipoprotein E in lipid efflux, was also upregulated. And so these two findings have given us a very important handle into really delineate the pathway for excretion of toxic lipid out of the cells. We went on to look at neurofilament light chain in Gaucher disease type 1 versus Gaucher disease type 3, and that was clearly increased in age-matched controls, as well as glucosylsphingosine. And in adults, there was an increase in neurofilament light chain compared to age-matched healthy controls. And these adults are at increased risk of Parkinson's disease, Lewy body dementia. We found in our patients that actually another lipid is significantly increased, and this was revealed by this mass spec imaging of the whole brains. And this was a big surprise, but it turns out GBA enzyme deficiency affects co-regulated lipid pathways. And we believe that lysophosphatidyl-choline and related lipids are very important co-conspirators in neuronal injury. In patients, amazingly, apolipoprotein E is markedly increased in untreated patients, and its ROC curve is 0.92, probably one of the best biomarker characteristic. Not only that, apolipoprotein E is correlated with glucosylsphingosine. So these and other data, and we have a lot of other data on neurofilament light chain, which really provides a foundation for exploring neurofilament light chain as a biomarker and studying the context of glucosylsphingosine as well, which we are currently doing. So as I mentioned, through the work of Shiny Nair in my lab and our collaborators in Amsterdam, we have validated glucosylsphingosine as a compelling biomarker that is now used worldwide. And in many ways, this biomarker fulfills the Cox postulate, if you like. It is an osteoblast toxin and explains many of the bone phenotypes. It is an immunoactive antigen. Our collaboration with Shriganga Chandra in neurosciences have shown that glucosylsphingosine accelerates pathological alpha-synuclein species formation. It induces macrophage multinucleation. And in wild-type mice, when glucosylsphingosine, or LGL1 in short, is infused, it phenocopies Gaucher disease. It's correlated with liver enlargement, spleen enlargement, and these other biomarkers such as chitotriosidase, GPNMB, and after treatment it falls. And as I showed you, the ROC curve is less than 1. So these data led us to propose this model, that when the enzyme is defective, the glucosylceramide is converted to acid ceramidase, generating lysogl1, and that is pumped out by one of the lipid efflux transporters. Another data we have is that in the blood, it is circulating in the high-density lipoprotein particles. So I want to discuss this late onset Gaucher neurodegeneration in particular. These are the manifestations of type 1 Gaucher disease. It's a multisystemic disease, largely attributed to this lipid-laden macrophages. It involves the liver, spleen, and bones. But in this type 1 patients, nearly 5% of patients are at risk of Parkinson's disease and Lewy body dementia. There's nearly 26% higher risk compared to the general population. So as I mentioned, there's huge phenotype diversity. I'm sorry, again, this my text did not appear on the left-hand side, but I'll talk you through the slide. But there is huge phenotype diversity as shown here. And even in homozygous L44P neuronopathic genotype, although as a group it is more severe compared to type 1, there is a huge variability. And in N370S homozygous, or these N370S Gaucher disease, there is again a huge phenotype diversity with first symptomatic presentation from childhood, sometimes into 7th or 8th decade of life. And so our premise is that neurodegeneration that we see in these types of Gaucher disease is a continuum to what we see in neurodegeneration in the type 1 Parkinson's disease. You might argue that Parkinson's disease is a sulfosynuclein mediated disease, and so it is. And it turns out that even in these early onset childhood neurodegeneration, alpha-synuclein pathology has been detected, but the predominant mode of injury is neuroinflammation and subsequent neurodegeneration. So we want to try and link these neuronopathic forms to the late onset neurodegeneration of Parkinson's disease and Lewy body dementia. So these are the clinical manifestations of Gaucher disease. And as I said, we detect it in the clinic by horizontal saccades when this normal movement in saccades is suppressed, and ultimately it leads to the patient just not able to move the eye. And then the head is just bobbing to follow objects. And by the time this happens, like this poor boy, really already neuronal injury is started, and that will undermine the full therapeutic potential of drugs that are coming up. And so we really must look for biomarkers before onset of disease. And so this is the data I thought I had missed out. But when we look at all of our mice and look at the neurofilament, remarkably, serum neurofilament light chain are correlated with serum glucosylsphingosine. And I showed you this other data of moving from mice to our patients in the clinic, showing that neurofilament light chain and glucosylsphingosine is elevated compared to age-matched GD1 controls. And also there is an age-related increase that we are seeing. So ApoE is an intriguing biomarker. I've already talked about this earlier, and we are pursuing this very vigorously. So in the next few minutes, I'm going to be talking about, can we use an inhibitor of glucosylceramide synthase and see what the effect is in the mice. And for sure, the neuronopathic Gaucher disease mice that live about 18 months live significantly longer, but the effect on survival is modest. And when there is a rescue of GBA in the microglia, this inhibitor can dramatically prolong the survival. And so replacing GBA in the microglia combined with substrate GCS inhibitor might be something that could be pursued in the future. Inhibition of GBA not only decrease the activation of the microglia, as shown here, in NGD mice, it is markedly increased, and these mice treated with the inhibitor have an improvement, decrease in microglia activation, as shown in this strain, as well as the microglia rescued mice. And what we find very interesting is that NK cell activation is also decreased. And so inhibiting glucosylceramide synthase enzyme could potentially have immunomodulatory roles beyond just substrate balance in Gaucher disease. And so these are the data which are entirely consistent with proposing neurofilament light chain as a biomarker, that these improvements in survival are exactly mirrored by reduction of neurofilament light chain from 2,000-fold elevated to only 100-fold elevated when microglia are rescued. And when the neurons are rescued, neurofilament light chain is only 10-fold elevated. And this is mirrored by glucosylsphingosine. And importantly, this is mirrored by immune dysregulation. So improvement of microglia and decrease of CCR2 positive macrophages and immune infiltrates in the brain. So I want to, in the last few minutes, talk about these mice, these late onset mice. And we found that these mice not only exhibited whole brain accumulation of sphingolipids, and they had increased circulatory glucosylsphingosine, and there is increased neuroinflammation with aging. And so these are the UMAP plots in these mice showing clustering of microglia from young and aged GBA flox mice that had CX3CR1 Cre and immune cells. Cells are clustered by as shown on the left and on the right. And the one thing that I want to draw your attention to is this subset of the microglia. And these are the these microglia are expressing methoxin 1 and thrombospondin 3, and this is these genes are contiguous to GBA1, and so I think that this locus beyond GBA gene itself might be playing an important role. So overall inhibition of GCS reverse damage associated microglia restored homeostatic microglia, and reversed NK cell activation, and also improved the lipidomic profile. So this is the summary before the last part on the modifier genes, that this is the picture model we've developed of neuronopathic Gaucher disease involving damage-associated microglia and border-associated microglia, leading to influx of CCR2 positive macrophages, cytokine secretion, macrophage activation, and neurodegeneration. When GBA is rescued in the microglia, there is an attrition of this pathway, including attrition of NK cells, and there is a reduction of neurofilament light chain, lipids, and ApoE. And when the microglia are rescued together with inhibition of GCS inhibitor, this entire pathway can be abrogated. So in the last few slides, please bear with me while I talk about the modifier genes. I talked about the modifier genes, a slide that you've seen before, and our basic premise is that neurodegeneration is a continuum from childhood onset to adult onset with different burdens of alpha-synuclein pathology, but the link between the two might be neuroinflammation. So here we have really delved deeply into the GBA locus as shown here, and this is a huge challenge in medicine. There are lots of papers coming out because of the interest of GBA in Parkinson's disease, how to reliably genotype this locus. And it's very complicated because of the pseudogene and highly homologous and the pseudogene for GBA1. So we've used a combination of techniques. This is work that has been done by Jeff Ruan and Dijian in YCGA under direction of Hongyu Zhao and Shrikant Mane. And PacBio third generation sequencing has helped us to really do long read sequencing. And then we've also validated that with Sanger sequencing. But every one of our patients have undergone whole exome sequencing. And so we have 550 Gaucher exomes, majority from Yale, but we also have collaboration from Argentina and Egypt. So I talked about ApoE. And so the nice thing about banking the genomes of patients that we care for is, as our understanding increases for any disease, we can go to that to really understand patterns of disease and genetic risk factors. And this is a brilliant example of that, because rather than doing simple chi-square case control studies, we can look at the whole exome data and use mixed effects models to see what is the odds ratio of this neurodegenerative promoting allele ApoE4 compared to Parkinson's disease, Lewy body dementia, and Gaucher disease. And in our cohort, we find that it has an odds ratio of 7. And conversely, ApoE2, which has been shown in Alzheimer's disease to be protective of Alzheimer's disease, is a significant protective factor also in the Gaucher disease patient population, very highly significant. And so this ApoE, as I mentioned to you, is a lipid transporting protein. It is the main cholesterol delivering protein in the brain, and the astrocyte is like a hepatocyte. And so neurons and all other cells need a lot of cholesterol, so astrocytes are making a lot of cholesterol, and that ApoE, lipidated ApoE, is effluxed out of the astrocytes through the ABCA1 transporter. This transporter is renowned for a very rare disease called Tengier's disease in Tangier Island in Chesapeake Bay. But we were interested in looking whether the ApoE variants that we saw somehow were also linked with ABCA1, and whether there were common variants or rare variants that are overrepresented in our Gaucher disease, Parkinson disease, Lewy dementia population. And what we found remarkably is that there were some common variants that were overrepresented in the Gaucher disease, Parkinson disease population with an odds ratio of nearly 6.57. And there were some variants in the untranslated region, which were also overrepresented in the Gaucher disease patients who went on to develop Parkinson disease, Lewy body dementia. And so this is my last slide. And in conclusion, I think that we have a lot to do. And together with our collaborators, we are moving forward to further define the significance of microglial GBA in neuronopathic Gaucher disease, pathophysiology, immune landscape, its role in different neuronal cell types, and really look for modifier genes such as in apolipoprotein E, ABCA1. We will clearly be doing iPSC cell works to really provide functional rationale as well as animal models. And as shown in a remarkable paper in Science a few weeks ago, we are also exploring GPNMB, which incidentally ours was the first group to identify as a biomarker and validate it in our mouse models. So here are my colleagues, and I apologize, I missed out Dr. Shriganga Chandra in neuroscience, who has been a very important collaborator in our studies. My group shown here, and then colleagues in Yale Center for Genome Analysis, as well as Kathy Klinger at Sanofi Translational Sciences. So with that, I thank you for your attention.
Great, thanks, Dr. Mistry, for this fascinating story. Now it's open for the question or comments. You can unmute yourself, and if you have question or comments. I'm gonna start into if no one have comments, starting asking you first. So for the microglia specific query, I don't remember when does the gene, the query expression pattern, like how early the expression starting? Have you tried to manipulate a sort of a different time point later, see if see the difference sort of the phenotypic causality?
So Dr. Jiang, that's a really key question because we generally think of microglia just like tissue macrophages. And it turns out that in embryonic development, the first cell type that start to populate the embryonic brain is microglia. So there is microglia plating. That is the first thing, even almost before neurons start to get established. And these microglia ontogeny has been traced to the yolk cell, right?
Yeah.
But the tissue macrophages are arising from hematopoietic precursors. So CX3CR1 allows us to knock out genes in cell types that are really arising from the yolk cell. Although I think the caveat to that, Dr. Jiang, is that there can be some CX3CR positive macrophages in the rest of the circulation, but majority, it is a very minor population.
As a follow-up for the modified exon you have done, so you show the 3 candidate gene. So that's all 3, or you have additional sort of modified candidate remain to be sort of investigated?
Absolutely, Dr. Jiang. And what our premise is that, you know, I used to talk to Dr. Lifton that I really want to identify modifier genes. And he said to me, listen, go away, get 1,000 patients and come back to me. As you know, we are never going to get 1,000 patients. And I don't think that inborn errors of metabolism lend itself to that kind of GWAS kind of studies, because the phenotype, every patient has a unique phenotype.
Yeah.
So, to do that kind of unbiased analysis is not a productive exercise. I think what we do is, as we delineate the phenotypes, then there are clear subgroups of phenotypes. And then applying something like mixed models, because these phenotypes are correlated with liver volume, spleen volume, and everything else, biomarkers, one needs to do this kind of multiple logistic mixed model analysis, because the numbers really go attrition. So in our cohort, for example, we've got only 11 patients out of 300 who've gone on to develop Gaucher disease, Parkinson. And this kind of challenge, which applies to all inborn errors, does not lend itself to, there is no way we are going to have 500 Gaucher disease, Parkinson disease patients. Mm-hmm. And it doesn't also allow us to do simple chi-square testing. Mm-hmm. The modeling has to be very different. And I'm very lucky that in my group, I have Mohsen Basiri, one of our lysosomal disease fellow, who has worked exceedingly hard for the last year with a group of statisticians and data scientists that I think is developing, in my opinion, unless someone can challenge me, a new model of how we should be looking for modifier genes.
Yeah,
in heterogeneous disease,
yeah,
in heterogeneous Mendelian diseases.
Yeah, that's definitely absolutely challenging for the rare disease. Anyone have any other comments or question?
Yeah, thank you so much, Dr. Mistry, for such an excellent talk. I was just wondering, like, due to such large phenotypic variability in the onset of the disease, that probably is also correlated with the biomarker detection. How do you envision diagnosing the disease in time for intervention?
Neru, thank you for that question. And this is a topic that has preoccupied many of the leading investigators. And, the first biomarker that was discovered that, Michelle Spencer and I have used quite a bit and discussed previously is chitotriosidase, described for the first time nearly 20 years ago. And it definitely correlates with disease severity, but the chitotriosidase gene is highly polymorphic, with 6% of any population that have a homozygous null variant. It does not impact Gaucher disease at all, but this individual do not express chitotriosidase. So they have zero value. So when in the era when we only had chitotriosidase, it was not possible to compare one population or another. And to your point, you are absolutely right that if a patient does not have the null variant, we can track them longitudinally. And so we are using that approach in our patient, but we don't just rely on that one biomarker. We look at a basket of biomarkers. We want to look at the biomarkers like glucosylsphingosine. I mentioned to you that I like to think of it as fulfilling the Cox postulates. That is a very important biomarker. But also, when we are looking at a child, we want to look at simple things like what is the growth parameters? How is the child doing? What is the spleen volume? What is the platelet count? And so it is a holistic view where biomarkers have a definite place. And and I'm sure Michelle Spencer is much like this. She looks at all of this data, and based on our knowledge of the field, we come out with a decision that here is a disease that is progressing and we must get on. What Mohsen Basiri in my lab is doing, he's developing an AI algorithm to capture the way we think about it, to see if we can develop some tools And such a tool would definitely, to your point, include biomarkers.
Thank you for the answer.
Great. I wonder, we already 3 minutes past 15. Any short question, or just remind everyone, if you need a CME credit, that's the chat, that's the phone number, the code that you need to take and type in right now. Otherwise, you're going to miss opportunity. If no further questions, thank you, Dr. Mistry, for taking your time presenting to us this fascinating story. Just remind you, next week we will not have a grand run. We're going to using that slot for our internal resident review. So we'll see you the week after. Thanks again for everyone joining.